Hello everyone, this is our second to last lecture for the semester, and it's a mini lecture. So to reflect that, we only have two main learning objectives. First, we're going to walk through the nitrogen cycle. We're not going to delve nearly as deep into it as we did for, say, glycolysis or the Krebs cycle. And finally, we're going to try to create two mnemonics to memorize nucleotide structures. Alright, the nitrogen cycle. As you probably know, humans and plants and animals are mostly made out of carbon, oxygen, hydrogen, and nitrogen. So how do we get nitrogen from the atmosphere into, I guess you would call it our biosphere, plants and animals? Well, atmospheric nitrogen is converted to stable products by bacteria, and these are usually symbiotic with plants. So once this gets into plants, well, animals can eat it. So nitrogen from the atmosphere eventually gets into animals, gets into us. Another relevant way, which I haven't mentioned here, is through the Haber process, which is the chemical process by which atmospheric nitrogen is reacted with hydrogen to form ammonia for fertilizer. So that's sort of a chemical industrial way to get nitrogen out of the atmosphere and into plants. So that nitrogen will go into plants and animals and bacteria as ammonia and products of that, and it'll get converted to nitrites and nitrates. So the oxidation to nitrites and nitrates goes through a series of reactions called nitrification. Really good name, really evocative. But eventually, as it's used, they can be converted back into nitrogen gas called denitrification. So nitrogen from the atmosphere goes through bacteria or the chemical industry into plants, animals, bacteria again, and to useful nitrogen-containing compounds, and eventually back to nitrogen gas. So in humans, nitrogen is really relevant for the urea cycle. So excess nitrogen eventually gets excreted as urea. So this is formed from amino acids and ammonia. As you know, amino acids have to have nitrogen in them. So that's one source of perhaps excess nitrogen in our diet. And this urea cycle occurs mainly in the liver. Number one, best organ. Anytime I've got a question for you, which is where does this happen, at least 50% of the time, maybe more, it's going to be the liver. Just number one, best organ all around. So the urea cycle is really tied into gluconeogenesis. Well, why might that be? Well, proteins, which contain nitrogen, are used as energy sources under starvation conditions when there's not a lot of carbohydrates or fats available to be used. But remember, you still need to make glucose to feed your brain, or at least make ketone bodies to feed your brain. So in that case, proteins would be used to chop them up to make new glucose molecules, and the nitrogen in that pathway gets excreted as urea. So let's talk about what happens to excess nitrogen within the body. So nitrogen is collected within the mitochondria of cells, and the carriers for that are going to be aspartate and citrulline. So here on the bottom left, I have aspartic acid. Recall that if we take off the protons, we'll end up with aspartate and citrulline. So you'll notice they each have nitrogen in them, so they need to be nitrogen-containing species in order to transport nitrogen, obviously. Alright, so here's sort of the figure we're going to use for the overall pathway. So on the left is the mitochondria, and on the right is outside the mitochondria in the cytosol. So we've already established that aspartate and citrulline are the carriers to move the nitrogen within the mitochondria out to the cytosol. So you can see those right here, aspartate and citrulline. But in the cytosol, the products of the cycle, there's going to be two of them. First, fumarate, right here, which gets used to make for glucose synthesis, gluconeogenesis. And finally, excess urea, which will eventually go to the kidney and be excreted in urine. Okay, let's do a quick walkthrough of the urea cycle. So starting in the mitochondria with excess nitrogen, which is represented here as ammonia. So ammonia and bicarbonate and a little bit of ATP will react to form something called carbamoyl phosphate. The phosphate is then displaced by the amino group of ornithine, which eventually forms citrulline. This is a lot of fun words. You don't really need to know the exact structures of these, other than just sort of how they take excess nitrogen out of the mitochondria and excrete it as urea and fumarate for glucose synthesis. So just to recap the mitochondrial steps, ammonia plus bicarbonate and ATP form carbamoyl phosphate, which then becomes citrulline through a reaction with ornithine. So remember, citrulline and aspartate are directly transported from the mitochondria to the cytosol. And there, an enzyme called argininosuccinate synthase, say that 5 times fast, catalyzes the condensation of these two together to form argininosuccinate. Argininosuccinate synthase synthesizes argininosuccinate. Okay, but argininosuccinate lyase hydrolyzes this molecule to form arginine and fumarate. So we already know that fumarate gets exported to participate in making new glucose molecules. So great, fumarate is done. It is out of the cycle and it's ready to go do its work. But we still have arginine left over. Oh yeah, so I guess fumarate, we're not quite done with fumarate yet. Fumarate gets converted to malate, which then ends up getting converted to glucose. Whereas arginine gets hydrolyzed to form urea and ornithine. And, well, you might recognize that word. Ornithine goes back into the mitochondria to restart the cycle, whereas urea is excreted. This is why it's called the urea cycle. You start off with excess nitrogen, shown here as ammonia, on the left in the mitochondria. Goes through a series of reactions to form a useful byproduct, fumarate, and a waste product, urea. And what's left over goes back into the mitochondria to start the cycle over again. So how do we regulate this? How does our body know when it needs to turn on the urea cycle? So most of these reactions are near equilibrium reactions. So the easy way to regulate this is just regulate the amount of nitrogen entering the cycle. Just regulate the amount of substrate. So because none of these reactions are really, they're all close to equilibrium, if we have excess nitrogen, it very quickly gets converted to urea. However, we can also induce the enzymes in this cycle. So if you're fasting, if you haven't eaten for a while, more protein will be broken down. So more protein breakdown means more excess nitrogen, which, as we just said, increasing the amount of substrate will increase urea. Also, glucagon, one of its many jobs, is to stimulate the liver to increase the synthesis of the enzymes in the urea cycle. So remember, the enzymes aren't actually being controlled, so to speak, but the amount of substrate is, and the amount of enzyme is as well. So it's not a very fine control, but on the other hand, we don't really need it to be a very fine-tuned control like a glycolysis does. Okay, so that's the end of the first half. That's what happens to where we get nitrogen and what happens to excess nitrogen in the body. So most nitrogen in the body is present as amino acids and proteins. So, that's sort of an easy thing to say. There's a lot of amino acids and a lot of proteins in your body. So, don't sleep on nitrogen and amino acids. But also, importantly, these amino acids are involved in the metabolism of lots of other useful things in your body that are not proteins. So especially important are nucleic acids. So we've already run into these a few times. ATP and GTP can be used as energy sources, and these are derivatives of nucleic acids. they can be cofactors for enzymatic reactions, and perhaps most importantly, they can be used for DNA and RNA. And here's what they look like. These are nucleotides, so they form the base of DNA and RNA. So on the top we have purines, we have adenine on the left and guanine on the right, and on the bottom we have pyrimidines, uracil, thymine, and cytosine. So first off, the major difference that you can see between purines and pyrimidines are that purines have 2 rings and pyrimidines have 1 ring. So the way I've always remembered this since I was in high school, actually, is something that Dr. Brent Richards at OSSM taught me, which is that purines are pure of heart. It's a little bit silly, but I guarantee you'll remember it for the rest of your life that way. Purines have 2 rings, similar to how a heart has 2 lobes, I suppose. So adenine and guanine have 2 rings, they're pure of heart. Whereas uracil, thymine, and cytosine only have 1 ring. They don't look like a heart at all. So you can just remember purines are pure of heart, they've got 2 rings. Pyrimidines aren't, they've only got 1. And if you want to take it even further, you could say you're gaga for the pure of heart. Gaga, guanine, adenine. That way you'll never forget which ones are purines and which ones are pyrimidines. So when I was making these slides, I sent a little thank you note to Dr. Richards to say, hey, wow, I still remember this, and I probably will until the day I die. And he said, that's great. I was actually taught this by Dr. Ann Dell, also at OSSM. So the chain of this mnemonic sort of exists for 4 generations now. So if any of you guys ever end up teaching biochem to someone, if you use this mnemonic, you'll extend this chain of knowledge. And I think that's pretty cool. Okay, but unfortunately this isn't high school biochem. We don't just need to remember what the purines and pyrimidines are. We also need to remember the differences within the purines and pyrimidines. We'll talk more about their functions next week, but for now let's just try to nail down the differences. So let's look at adenine and guanine. So if we look at the numbering here, starting with 1, position 1, 2, 3, 4, 5, 6, you'll see that the only positions that differ between adenine and guanine, the purines, are positions 1, 2, and 6. So on adenine, this carbon doesn't have any substituents other than hydrogen, whereas on guanine, we have an amino group right here. At position 1, this nitrogen has a double bond to position 6 on adenine, whereas it's just a free amino group in guanine. Finally, adenine's position 6 is single bonded to an amino group, whereas guanine's position 6 is double bonded to an oxygen. And you'll note that all the other positions are the same. The smaller ring, the 5-membered ring, positions 4, 5, 7, 8, and 9, that's exactly the same between the two. Also, position 3 is the same between the two. So take a few minutes, pause the video here, and think of a mnemonic to help you remember the structure of purines. What I would recommend is memorizing the structure of one of these, say guanine. Memorize how to draw it, and then think of a mnemonic to help you remember that positions 1, 2, and 6 are different, and how. So basically, if you can draw guanine, figure out a way to help you remember what the structure of adenine is. So take 5 or 10 minutes, work that out. I guarantee you, I'm going to ask you your mnemonic on a quiz. So, yeah. Okay, let's do it again, but for the pyrimidines. So if we look really closely, we'll see that the pyrimidines, they only have 1 ring, so they should be theoretically easier to remember. But there's 3 of them. There's uracil, thymine, and cytosine. But they only differ at positions 3, 4, and 5. So let's focus on the difference between cytosine and thymine. Position 3 on thymine, single bond to the carbon on position 4. Position 3 on the cytosine, double bond to the carbon at position 4. Okay. Position 4 on thymine, carbon double bonded to oxygen, right here. On cytosine, carbon single bonded to nitrogen. This is similar to the difference between adenine and guanine, if you'll recall. Finally, between thymine and cytosine, the only difference is that thymine has a methyl group on position 5, whereas cytosine just has a hydrogen. So 3 differences between those. But if you can remember the difference between adenine and guanine, I guarantee you can figure out the differences between cytosine and thymine. And here is the trick for the mnemonics for the pyrimidines. If you can memorize thymine, it's exactly the same as uracil, but it doesn't have anything on carbon number 5 there. Exactly the same. So I think it's really helpful when you're memorizing these to sort of have a flow. If you can memorize thymine, how is thymine different from cytosine? If you can memorize thymine, how is thymine different from uracil? So take 5 or 10 minutes, draw these a few times, get used to their structure, and think of a mnemonic for how you might help yourself remember these down the line, say 10 or 20 years later. Alright, so one more quick thing before we finish this lecture. As you might know, DNA is short for deoxyribonucleic acids. So what's the deoxy in there for? So we know that ribose is a sugar. We learned that all the way back in chapter 4. So a deoxy sugar is going to be a sugar where one of the hydroxy groups is replaced by a hydrogen, highlighted here in red. So if you start off with these riboses, perhaps from the pentose phosphate shunt, as we learned a week or two ago, the removal of this hydroxy group to just the hydrogen is catalyzed by ribonucleotide reductase. So we don't just start with these deoxy sugars, this deoxyribose. We have to manufacture it. Alright, so we'll talk more about the function of the nucleic acids in the next lecture. So here's a summary of our nitrogen lecture for today. So the nitrogen cycle occurs in the mitochondria and cytosol of cells in the liver, and the input is basically waste nitrogen, and the outputs are urea and also eventually products that can be made back into glucose. So purines, G and A, are pure of heart. They have 2 rings, and they differ at positions 1, 2, and 6. Whereas pyrimidines, C, T, and U, are not. So they only have 1 ring, not 2 rings. And they differ at positions 3, 4, and 5. And alright, that's the end of this 20-minute lecture. If you have any questions, let me know. And see you in the last lecture next time for nucleic acids.